Hello everyone, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we're going to be talking about how host and microbes interact, particularly the human host. In the last couple of videos, we've been talking about the immune system, both the innate and adaptive immune system. If you haven't seen those videos, highly recommend check those out before you watch this one. But today, we're going to be focusing on how microbes interact with the human body. So first thing to consider, we do have some terms that we use to describe the types of interactions that happen between microbes and humans. And these interactions are symbiotic. And a symbiotic relationship just means that two organisms are interacting in some way. So the first type of relationship, a mutualistic relationship, is demonstrated here. Bacteria, normal flora that live in the gut, in the large intestine. And they benefit because they're getting a constant supply of food from us and really stable environmental parameters, temperature, pH, osmotic pressure. What do we get out of the deal? Microbes will produce vitamins for us. They take up binding sites, which reduce the likelihood that pathogens are going to be able to colonize. They produce antimicrobial chemicals that, again, will target pathogens. They prime the immune system, which keeps our immune system healthy. And there's a lot of other ways that the normal flora, the normal microbiota, assist us human beings. The next example of a symbiotic relationship is that of a commensal relationship. And in a commensal relationship, one of the organisms is benefiting and the other is basically unharmed, but they're not really helped. And so an example would be the microbes that live on the surface of our skin. These microbes, again, they're getting a food source, they're getting a surface area to colonize, they're getting relatively consistent environmental parameters. And for us, it doesn't really hurt us, doesn't harm us. We're basically indifferent to them on the surface of our skin. Now, the relationship that we tend to be most concerned about is the parasitic relationship. And in a parasitic relationship, one organism is benefiting at the expense of another. And so you could think about microbes that cause wound infections. Yes, they are consuming organic compounds that is human flesh, and they are getting that stable environmental parameters, but they're also producing toxins and they're damaging our tissues. And so we're being harmed at the expense of the microbes. And that's an example of a parasitic relationship. Now, when it comes to parasitic relationships or infections, there can be a few different types. There can be a primary infection, and we've talked about that in videos in the past where a microbe establishes itself, it attaches, it colonizes, and causes some type of harm to its human host. But there are also opportunistic and secondary infections. And there's a little bit of a difference between the two. A secondary infection relies on a primary infection in order to colonize. And so that primary infection had to be there first. It could be viral, could be bacterial, in order for the secondary infection, the secondary organism, to establish itself. An opportunistic infection is a little bit different because an opportunistic infection is relying upon a weakened immune system in order to establish itself. And so that could be a microbe that establishes itself as a result of the host, a human being, going through chemotherapy that is destroying the host immune system. Could be HIV. Now, HIV gets a little tricky because the microbes that establish themselves as a result of an HIV infection, well, HIV is also weakening the immune system. So it could be a secondary and an opportunistic infection in that case. But the point is, the difference is secondary infections rely on a primary infection in order to establish themselves. Opportunistic infections rely on a host's weakened immune system in order to cause infection. And when there is an infection, we know that microbes first have to adhere to their host. Without the adherence, the attachment, it's really hard to cause an infection. And so adherence usually happens first, or attachment. And then there is the colonization, the microbes growing and dividing in or on the host. The microbes may then at some point invade the host tissues, cross over a physical barrier if they haven't already, and they may also do what's known as delivering effector molecules. They can actually create chemicals that make it easier for the microbe to establish itself in the human body. When the microbe is attaching and colonizing the human host, this is often the incubation period where the microbe is growing in concentration, and during that time, us human beings, well, we're starting to show the signs and feel the symptoms. And what's the difference between the two? Well, if we're showing signs, signs are things that we can measure. And so that would be a fever, blood pressure. So it's something that we can actually measure. The symptoms are what we're feeling. I feel tired. My body aches. malaise, that kind of thing. And so during that incubation period, we're starting to get sick. That's also the time the immune system is beginning to encounter that pathogen and beginning to mount a response. And so we have an illness phase, incubation, then illness. We might be, if it's a respiratory virus, sneezing and coughing. If it's influenza, maybe there's a fever involved as well. But then as the immune system begins to hopefully win that battle, we have the convalescence phase. And the convalescence phase is kind of like a phase of healing where the immune system beats back the virus and we start to repair the damage and feel better. Now, at that time and beyond, we could still potentially be carriers of that particular pathogen. So we could still have enough of that pathogen to be carriers and potentially spread it to other people. Usually the most likely time that we're going to spread an infection is during that illness phase. Now, if an infection ever winds up in the blood, we call that an emia, ends in E-M-I-A. And so if there is an emia, that means something is in the blood. And so there could be bacteremia, where there are bacteria in the blood, viremia, where there are viruses in the blood, septicemia, which means we have a systemic infection that may include bacteria, parts of cells, and waste components. And so once something winds up in the blood, it can potentially be a little bit more serious, especially if we're talking about bacteria or some of the toxins they produce. And speaking of toxins, there are a couple different types of bacterial toxins that are really concerning for us human beings. The first being an exotoxin, which is a protein that is usually produced either secreted or excreted by a bacterial cell. Again, it is a protein, which means it is going to be impacted by heat. And sometimes you can use these as toxoids and create a vaccine that will train the body, train the immune system on how to target them. But these can be very serious and they can cause severe damage to humans. And when there is toxemia, toxins in the blood due to exotoxins, that can be a serious thing. There's a couple major types of exotoxins, one of which is the AB toxin. It's actually two parts. The A is the part that is active, and the B part is the part that binds to cells. And this AB toxin can cause damage, especially to epithelial cells. The other type of exotoxin that we are somewhat concerned about is something called a superantigen. And what the superantigen does is it actually binds immune system cells together. And you think, well, don't they usually communicate that way? Yeah. But what the superantigen does is it will bind, let's say, a helper T cell to some type of antigen-presenting cell, but it keeps them together for far too long. And in the process of doing that, we get way more pro-inflammatory cytokines than what was intended, and that can cause all kinds of problems throughout the human body. The other type of toxin, and it's probably even more concerning, is the endotoxin. And the endotoxin is actually the LPS part of a gram-negative cell wall. And this stuff is really, really reactive. The human immune system cannot stand LPS in the body, and it tends to freak out a lot about that. And when we get endotoxin in the blood, that can cause things like septic shock. Now, when we're talking about shock here, medical shock, it's not the same thing as, wow, I got an A on that test, I was shocked and I had to sit down. No, that's not what we're talking about here. Medical shock, what winds up happening is we talked about the 5 cardinal signs of inflammation. If you remember, we said that one of the things that happens is the blood vessels dilate heading towards the wound so that we can get more immune system cells to the wound, and they constrict away from the wound. Well, if you have endotoxin throughout the entire body, essentially your entire body is infected, well, that causes a release of cytokines that are pro-inflammatory all over the body, and all the blood vessels dilate. Well, if all the blood vessels dilate and you have a fixed volume of blood, what do you think is going to happen? Blood pressure is going to drop a lot, and that can cause life-threatening illness because now the brain, the heart, the liver, the kidneys, they're not getting enough oxygen. So it's a very, very serious condition. And that's why when we have an emia, when we have bacteria, and especially septicemia or toxemia, something in the blood, that can be a very serious medical condition that needs immediate medical attention. Perhaps one of the most frustrating things when it comes to host microbe interactions, especially when we're dealing with infectious agents and especially bacteria, is they have sneaky ways to avoid the immune system. If the immune system worked perfectly all the time, that would be great. But we know through human experience that that sometimes isn't the case. And so what can these microbes do to avoid the immune response? Well, thing number one, we've got a picture of an antibody here, and one of the things that some microbes can do is they can actually break them down. They can break down these proteins, and when you break down the antibodies, they can't do the 6 protective outcomes we talked about in the previous video. They can potentially, and this is very strange, but some microbes can actually flip the antibody backwards. And so we learned in the last video that antibodies, the Y region, binds to specific epitopes on the surface of an antigen. And the tail is supposed to stick out, and that's the constant region. Some microbes flip that around, and it's the constant region that binds to the antigen. The Y part is sticking out. Well, the immune system cells can't recognize that. And so they render the antibodies mostly useless. There are certain microbes that can actually do damage to immune system cells, like our macrophages and our dendritic cells and T cells and B cells. There are some microbes, and we've talked about these types of microbes throughout the semester, that actually live inside of immune system cells and in fact require them in order to replicate. So we talked about things like tuberculosis. We've talked about Shigella in the past that actually depend upon the human immune system cells as sort of a home. Perhaps one of the most devious things that microbes can do, some microbes, and especially some multicellular parasites, they can change the way they look. It's called antigenic variation. And so what they do is they take the epitopes on the surface of their cell or on the surface of their body, if it's multicellular, and they change them constantly. And we know now by studying the adaptive immune response that it's highly specific. And so we make these antibodies that are specific for a particular epitope on the surface of an antigen. And some of these microbes have genetic code that is dedicated to changing these proteins so that the antibodies and variable regions no longer recognize those proteins. And they just keep doing this. There are some parasites, some infectious worms, parasitic worms, that spend their whole lives doing this, and they can actually establish themselves in the human for decades because they're constantly changing the way they look, and the immune system can never fully recognize them. You know, something else to consider, sometimes our own immune system can do damage to us just by trying to fight an infection. And so sometimes our immune system is responsible for a lot of the signs and symptoms that we feel. I mean, if you think about it, we feel really lousy sometimes because we have a fever. Well, what caused the fever? Yes, the infectious agent got into our body, but it's our body responding to that infection. The coughing, the sneezing, why is the extra mucus there? It's our body trying to flush out those microbes. And so that's the reason for the congestion. But there are other things that can make us miserable too long term. And so things like autoimmune disease, sometimes our immune system doesn't respond to antigen properly. And so what winds up happening is it actually thinks our own body proteins, the proteins on the surface of our cells, are foreign and starts to attack that. There are times when the immune system produces antibodies that will attach to an antigen, and our bodies cannot clear these antibody-antigen complexes that causes chronic inflammation. And with that chronic inflammation, sometimes we get a lot of complement protein that is produced, and we learn that complement, one of the things that it can do is cause cell lysis. And when that happens, it doesn't just lyse bacterial cells, it can also lyse our own cells once the safety mechanisms on the surface of our cells are saturated. Now we can also experience that cell lysis as well. And of course, we have abnormal responses like allergies, where the immune system thinks that things like cat dander and pollen and dust and milk and proteins from nuts are foreign and dangerous, and sometimes they're just not. All right, everybody, so that was just a quick overview of some of the aspects of host-microbe interactions. As always, thank you so much for being here. Have a great week.